Let's take 2 minutes and look at the structure of double-stranded DNA. We're going to find the structure of double-stranded DNA in the Protein Data Bank, or PDB. It's Protein Data Bank, but it actually houses a lot of different biological macromolecules like proteins and nucleic acids. So in the Google, just go ahead and type in B form of DNA, or bDNA for short, and then let's make sure we are directed to the PDB, the Protein Data Bank. There's a couple of entries. I recognize the second one is a nice tutorial. The PDB puts out a number of tutorials, and PDB 101 is going to send us to one of those tutorials. There you can see a couple of forms of bDNA and a zDNA form. Let's select that middle structure, and that brings up the record for 3BSE, and let's download that file. And so now I have a file of that particular structure on my desktop in a file form that's a text file, and so I'll just dump that into my text editor on the Macintosh, and there you can see the file. It looks like a bunch of lines of code almost, but there's some atoms and then numbers. Those numbers correspond to the atomic coordinates for each atom, atom 1 through the hundreds of atoms that are there, so X, Y, and Z coordinates. Highlight that text and just drag it into the Jmol window. And then in Jmol, we can hide those red dots. Those are just water that came with the crystal structure, so you can get rid of those, and now we can look at the backbone so you can more clearly see the double-stranded DNA. There it is. Let's rotate that and get a nice smooth rotation going on. Let's select each of the nucleic acids. First, guanine, G. Select G and color it green. Now let's select its complement, cytosine, and color that yellow. And now the last two bases, A, and color the adenine red and the thymine blue. And now what you can see is all of the greens and yellows match up, and all of the reds and blues match up, the GC-AT pairing. And so we've got the perfect complements of those nucleobases in the center of that double-stranded DNA.